All right, welcome everyone. Um, I'm sure more participants will continue rolling in. I'll just start the introductions. So I am Olivia Carmo. I'm a postdoc studying anhydrobiosis in the Boynham's lab at the Baylor College of Medicine. And today I'm stepping in for Joanna Murray to introduce today's speaker, Matt Stata. But first, I should mention that this webinar is hosted by the Water and Life Interface Institute, or WALI, which is an NSF-funded consortia of labs across the US studying anhydrobiosis. Um, and apologies, I don't have my WALI background or the usual WALI slide that Elena would post. But some rules for today's talk or best practices, or if you want to convey your questions, please submit those in the Q&A box. And Matt did mention to me earlier that there may be room for questions over the course of the talk. So if I see pertinent questions pop up in the Q&A box, I may ask them during the talk. So please feel free to submit questions over the course of the technical workshop.
And I'll try to pause for questions regularly too.
Perfect. Thank you so much, Matt. So now to introduce Matt. So Matt Stata got his PhD from the University of Toronto, where he studied C4, the evolution of C4 photosynthesis in Glipharis genus. And currently he's a postdoc in the Relab at Michigan State University in the Plant Resilience Institute, where he's worked on many things, including single-cell transcriptome studies of drought tolerance in C4 plants, in addition to studying the evolution of thermotolerance of a cool C4 eudicot that thrives in Death Valley. So, and just last year in 2025, Matt received a seed grant to study the evolution of C4 photosynthesis in two genera using a comparative approach, which is always super powerful. And in parallel to all of this, Matt has been working on various software tools, including UnigeneFinder, which is an amazing fully automated pipeline that simplifies gene prediction for de novo transcriptome assemblies, so you don't necessarily need the reference genome. And particularly exciting news is that Matt will be starting his own lab in the Department of Biology at the University of Massachusetts Amherst this autumn. So in that, Matt's lab will continue to study the evolution of C4 photosynthesis, and I'm sure develop many more very useful tools as well. So today's talk is actually a techniques talk for exploring the many applications of RNA-seq data. And again, just as a reminder, please put the questions in the Q&A box and I can ask them over the course of the talk and at the end. And with that, I'll pass it over to Matt. Thank you so much.
Great. All right. Thank you very much, Olivia, for the kind introduction. So as you're all aware from the information that went out that prompted you to sign up for this webinar, today I will be trying to give you a general overview of the various things that RNA-seq data is good for. And I will be providing a number of repurposable scripts in the form of slurm job scripts that can be run on a high performance compute cluster type system with nice, clear, edit the fields in this box to suit your own data sections near the top. And I'll talk a little bit about how you can repurpose those to your own systems. And also 3 Python scripts for doing some general expression analysis. I had planned to provide a lot more modifiable scripts for things like de novo transcriptome assembly and genome annotation using RNA-seq data, but those are pretty big topics, and it very quickly became clear that it would be difficult to provide a single script that could be easily repurposed to everyone's use cases. So for those sort of more advanced topics, I'm just going to sort of give a brief overview of the software tools that exist and describe how they work. And if anybody, was really hoping to hear more about the nitty-gritty details of that and get started with using those tools, feel free to reach out to me and I can provide assistance with that. But it seemed to be beyond the scope of this, and I wanted to keep this a manageable webinar that would give you really actionable information. So at the left of my slide here, this is sort of a table of contents style overview of everything I'm going to be talking about, and this will keep us oriented as I go. First, I'll cover quality control of FastQ read data. This will cover both uh, tools that can be used to generate QC reports yourself, but also these are the standard tools that are used by most sequencing companies. And when you get your sequencing data back, you will see information that's very similar to this in the reports that they give you. So even if you're not running these tools yourself, it's useful to know what they are telling you. Um, and then we'll get into tools for mapping reads onto a reference of some kind, whether that reference is a set of coding sequences or a full genome assembly, and calculating expression based on those data. And I'm going to just sort of provide an overview of the two tools that I use most frequently here in this section, and then we will come back to those later in the user repurposable scripts portion of the talk. Then these next three items are going to be touched on pretty briefly, but like I said, if anyone was, signed up for this webinar just because you wanted to learn about de novo transcriptome assembly and I don't cover it in enough detail, please reach out to me. So I'll be talking about reference-based assembly, which is when you have RNA-seq data and you do have a reference genome, but you nevertheless want to generate an assembly with your RNA-seq data, maybe because you suspect there are novel splice isoforms that may not be in your genome annotation. De novo transcriptome assembly, which is assembly of RNA-seq data in the complete absence of a reference genome. And then genome annotation, which is, for instance, if you had just sequenced and assembled a brand new genome and you don't have any gene models for it yet, RNA-seq data, along with protein databases for homology-based inference, are critical for that sort of genome annotation. Then I'm going to devote a good chunk of time to going over the scripts that I've shared. And when I get into this section, I've got a GitHub link for where you can get the scripts from. And then I'm hoping to leave some time at the end for discussion and Q&A. And also, I have in another window here, I've got a RNA-seq data read mapping pile up open in Genius that we can take an interactive look at if there's time and people are interested. So jumping right into quality control, this section will cover two software tools, mainly FastQC, which is what actually generates the quality report metrics for your RNA-seq data. And this FastQC is run on the sample level. You get one report per sample. And then multi-QC, I'm just going to touch on briefly at the end, although the slurm script I have for these tools does use both. Multi-QC is basically just a tool to aggregate the reports generated by FastQC if you've got a large collection of samples and you want to put them all in one nice centralized HTML file that you can flip through your samples and see comparisons of the various metrics and also other aggregate metrics across all of your samples. So FastQC, like I said, one report per FastQ file. It gives you our quality scores by base and read. So you have information about what is the distribution of quality scores across all, say, 150 base pairs of your reads. Sorry, I'm jumping through slides by accident here. It'll tell you things about the general base composition and the percentage GC content, which can vary from sample to sample or gene to gene. And it will give you information about the amount of duplicate reads, so reads that cover largely or completely the same sequence, as well as whether any sequencing adapter bits seem to be remaining in your reads that might need to be removed. And it'll provide you information on any sort of sequences that are overrepresented in your data, which could be the result of genes with very high expression. For example, in overexpression studies, if you're driving a gene of interest with something like a 35S constitutive promoter, we've seen recently that under the right circumstances in protoplasts, that can get you expression that's on the order of about 250,000 TPM, which basically means that 1 in every 4 transcripts is coming from that gene. So that would definitely show up as overrepresented and is expected and not a problem. And so the basic use for this is to screen for issues before any sort of downstream processing and to know what sort of lies ahead and what you might want to keep an eye out for. So next I'm going to go through, I ran FastQC on some of my own RNA-seq data. And these are some of the plots that it gives you in the HTML files that it generates. These are also likely the sort of plots you would find in a quality report that's provided to you by a sequencing provider like Novogene or other big name companies. So what's shown here, this is the distribution of quality scores across the 150 base pairs of a read. And the little blue line up at the top is sort of like a histogram of where not a histogram, sorry, but like the median quality value at that position across all reads in the library. So everything is nice and high up in the green section. This is what we like to see. There's a little bit of a decline as we head toward the 3' end here. And that is common in Illumina data, but it's still well within the high quality green region, so that's not a cause for alarm. And sorry, I can't see my own notes because I've got Zoom stuff in the way. There we go. And so trimming, if, for example, this tail end really was dropping off and the last little, maybe 10 base pairs had consistently low median quality, that would be a sort of a red flag that you might want to implement some kind of end-based quality trimming of your reads to remove that. This is one of the metrics that actually came up as a fail in my data. And then what this is showing here is this is the base composition at each read. So each of these different colored lines represents one of the 4 different nucleotides in not in RNA, but in cDNA, which is what's actually sequenced. So you have a T rather than a U. But what we like to see is sort of the same baseline across everything, which from about base 15 and on we get. But in the early end of the read, we get significant discrepancy from position to position. This is not automatically a cause for alarm, but it's nice to know that this is happening. And this can happen. This can happen because of non-random priming of the reads during library construction. Basically, it just means that the so-called random primers in a library prep kit don't all have exactly the same efficiency. So what's being primed at the end is not a true random distribution. This is not likely to cause problems in read mapping to a reference or in expression analysis. This might be something that you would want to watch out for in something that's much more sensitive to sequence anomalies like de novo transcriptome assembly. But yeah, this came up as a fail in my data set, but just because you get 1 or 2 of these metrics failing, if you know what they mean, you may or may not actually regard that as a reason to discard your sample. In this case, this is definitely not a reason to discard this sample. This is the GC distribution across all sequences. So this is a histogram now of mean GC content, and this is what we like to see, this sort of bell curve distribution. And this is another metric that my data actually failed. This is the percentage of sequences with a high degree of duplication. So sequences, reads with sequences that are identical to other reads. Under ideal circumstances, you'd want to see this line drop and then just kind of stay down. But this sort of rise here indicates that there is a substantial amount of reads that have duplicate sequences. This is not really a cause for alarm, particularly because in these leaf transcriptome data sets, there are a number of really highly expressed genes, which can result in identical reads or strongly overlapping reads. So again, this might be a cause for concern. And what this would lead you to watch out for is when you go ahead and map your reads to a reference, if you're getting a low percentage of your reads mapping, which the tools that I am going to cover for read mapping will give you a report of that. They'll say something like 80% of reads were assignable to the reference. Um, if you look in the reads that did not align, under situations like this where you have a high degree of duplication, you might find that these are things like weird primer dimers in your library prep and things like that. And so if you look in the reads that didn't align and find that there's a lot of duplication among those, that would point to this duplication being artifactual. And then this is a report of the percentage of adapter sequences that are in these reads. And so percentage in the y-axis, you can see that one of these lines, the red line representing the Illumina universal adapter, is creeping up toward the 3' end here to about 2%. So this can be a useful metric for telling you whether the sequencing company may have failed to adequately remove sequencing adapters. They usually do a pretty good job of this. And it can also tell you which type of adapter you might want to use for downstream read quality trimming and filtering to do additional removal of these adapters. So those are the main metrics that FastQC will give you. And then multi-QC, like I said, just aggregates FastQ outputs across samples, which is really good for comparing side-by-side patterns across your samples. Or also it generates a number of aggregate plots as well, where you can sort of see, for example, the GC content of your samples plotted for all of them. So you can see if mostly the lines all overlap, but one of them is skewed weirdly, then maybe that represents a sample that had something go wrong with it. It's good for identifying batch level patterns, and it's just really a convenient way to review a single consolidated report. And so again, the job script that I have that we'll come to in the second last section of this talk implements both FastQC and multi-QC, and you can really easily repurpose this to your own data and see the reports. So bringing it all together, what's the point of all this QC? Basically, the purpose of these reports is to guide any sort of pre-processing decisions that you may want to do. To inform you of whether you want to even bother with any sort of sequence-based, quality-based trimming and filtering of some of your reads. For expression analysis, it's usually not that critical, but for more sequence critical things like assembly, it can matter a lot. To compare your findings across your samples, and to basically determine how you will, like I said, how you will handle trimming and alignment. And critically, as I said already, a failed module, a failed metric in one of these reports is just something to review. Don't regard it as, ooh, one of my metrics came back with a fail. I better, do all my extractions and library preps again and send it off to the sequencing company and pay another $600 to get my samples all replaced. No, it's usually just something to be mindful of. And then finally, I just wanted to cover a few common tools that are used for quality-based filtering and trimming of reads. Trimmomatic is the tool that I've implemented a script that you can repurpose for your own data for. But the other two are commonly used tools that I just wanted to bring to your attention as well. So Trimmomatic, it trims based on quality from the ends of your reads and also uses a sliding window that scans across reads. And if the quality over that sliding window drops below a certain threshold, it removes that part and splits the read into multiple reads. And then if what remains is below a certain length threshold, it removes that. And it's particularly well suited for Illumina type paired end data. Cut adapt looks to find and remove sorry, I should also mention that it can be used to remove adapter sequences as well. And this is done by providing it with a FASTA file that contains the adapter sequences you want it to remove. Cut adapt also can remove adapter sequences. It can also find anchored or partial adapters, which is really nice. And it trims, again, low quality ends of reads, which is something it has in common with Trimmomatic. And it's got a great deal of flexibility for custom protocols. And then FastP is a tool that can be used for sort of an all-in-one to do the cleanup and also to give you QC reports in the style of FastQC. It auto detects adapters, which is nice, and again trims based on quality of the reads. And I should have mentioned this earlier, but for anyone who's not familiar with the structure of FastQ data, it's basically very similar to FASTA data, where you have an identifier and then a sequence for every sequence, in this case, every read. FastQ data has 4 lines instead of 2 for every read, and those extra 2 lines are, I honestly don't know the purpose of the third line. It's kind of a throwaway line, but the fourth line is a weird different ASCII character that indicates a quality score for each base. So your FOSS-U data has embedded right in it an assignment of how likely each base is to be true rather than a sequencing error. Okay, so that I think, yep, that's the end of my QC section. So if there's any questions, I don't know if there's anything in the chat or sorry, not the chat, the Q&A, don't put it in the chat. Is there anything there yet, or should I just
I don't see anything there, but I have
okay,
a couple of questions, if I may.
sure.
The so back to that plot where you had the adapter.
This guy?
Yes, this one.
Yeah.
Maybe you said this, but what's kind of the threshold at which you would maybe go back and do some trimming, not trimming, but removal of adapter sequences?
Um, it depends on your downstream. If all you want to do is map to a reference and quantify expression, most tools to do that are pretty robust. What they will do is if there's a read that would otherwise map beautifully onto a particular region of the reference, but it's got a little bit of adapter sequence at the end that doesn't map, these tools will generally just sort of soft clip that as they
Mm,
go. And it won't affect your ability uh, to quantify gene expression.
mhm,
Things like assembly tools are a lot more sensitive to those problems. So, for example, Trinity, which I'll cover briefly later, that does de novo transcriptome assembly,
mhm,
it essentially builds a big connected graph of, the nodes are not reads; they're what's called k-mers or sequences of
mhm,
length k that are in your reads, and then it puts edges between nodes if those overlap in the reads. And so if you've got an adapter sequence that is common and all over the place in your reads, that can lead to these sort of weird hubs in that assembly graph and result in all sorts of weird artifacts in the output. So for anything that's really sensitive to sequence and doesn't have a reference to check it against, I would almost always do some kind like this threshold. I would still do adapter removal for this if I was
okay.
doing de novo transcriptome assembly.
And it did seem like later when you're like for Trimmomatic and the other options,
Yeah.
there was already some adapter um, cleanup, I think.
Yeah, so
Yeah.
Trimmomatic, and I have a slurm job script for Trimmomatic, and I'll show you where the adapter file is provided there. And so that would be something that's in the user modifiable code block. So you could just say to your sequencing provider, what's the adapter file I should use for Trimmomatic, and then just swap that in there if they've used some kind of weird adapters. But the one that I've used is a pretty good start for most Illumina data.
Hmm.
And cutadapt and FastP I'm not so familiar with, but as I understand it, cutadapt needs to have I think it needs to have the adapter sequences provided as well, and then FastP tries to auto detect them, and I'm afraid I don't know exactly how that auto detection works. Those other two software tools I just sort of provided as points of comparison and other tools that people might want to explore. Trimmomatic is the one that I have used the most frequently.
Let's see. And how does all of this QC vary if it's long read RNA-seq, like iso-seq from PacBio?
Right. So I don't have I haven't actually done any iso-seq myself. I do know that PacBio is more prone to point errors. Um, but I think I think long read data like that has its own software tools, or at least its own suite of settings that you would want to
Hmm.
use for this. But yeah, this webinar focuses pretty much on just on
On short read.
NGS. Yeah,
Mhm. There is a question from Madison
sure.
Part. The MSU sequencing core is pivoting to more ABT sequencing, if
Yeah,
I'm pronouncing that correctly. Is Trimmomatic suitable for that? And do other
absolutely. Yeah.
aspects of QC change for the sequencing?
Absolutely. Yeah, the output from that is FastQ data exactly the same. It has the same what's called Fred with a PH, not an F. The same Fred encoding QC metrics in that fourth line for every read in your FastQ data. So Trimmomatic absolutely can be used for that too. The only thing I'm not sure of is what you would want to use for the FASTA file of adapter references that you provide. So Trimmomatic, when you install it, it comes with, I think, maybe a dozen FASTA files for common adapter sequence sets. What I've done in the past, just to be extra cautious, especially when I'm doing de novo assembly, is and also when I'm not sure what sequencing platform was used for the data, like it's data that I downloaded from somewhere and I have no idea what they did. What I've done to be extra conservative with adapter removal is I've actually just concatenated all of the reference adapters in the Trimmomatic folder and used the union set of all of them for adapter removal. Just to be absolutely certain that there was nothing funky going on there that could screw up my assembly. But that would be certainly something that Emily or Kevin, for those of you not at MSU, those are the folks in charge of the MSU Genomics core. That would certainly be something that you could just say, hey, if I want to do Trimmomatic on this, what are the adapter sequences I should be providing?
Fantastic. Thank you.
Cool. So the mapping and expression, this is going to be a major focus of this webinar, but I am going to just go over it somewhat briefly here, and then we'll come back to it in the user editable scripts toward the end. So the two tools that I use most frequently for this are HiSat2 and Salmon. So HiSat2 is a truly alignment-based read mapper that takes your reads and maps them onto a reference genome of some kind. Your reference genome could be, depending on what your purpose is, you could use just the coding sequences. Like you could, for example, download the CDS primary isoform only FOSTA file for a genome of interest off of Phytosome or your favorite genome repository. Or you could map onto the full length genome assembly where you're mapping it onto any possible base pair along all the chromosomes. And there's different reasons why you might want to do both. As you might imagine, mapping onto the full genome takes proportionally longer than just mapping onto the CDS, but both are reasonably quick. HiSat2 can perform splice-aware alignments across introns, which makes it particularly well suited to when you want to map onto the full genome assembly and see where your reads are spanning from one exon to another and to which exon in the case of multiple isoforms. The output from this is SAM format, which is easily convertible to BAM, which is just the binary equivalent. SAM is a text-based format that you could actually read with a text editor, although usually the results of this kind of mapping are like 12 gigabytes or so. So you probably don't want to try to open a 12 gigabyte file with Windows Notepad, but you could, for example, you could run the head command in a Linux terminal and see the top several lines of it. Or you could grep, which is the Linux search command for a particular sequence ID in it, and then BAM is just that, but converted to a more compact binary format. It's useful for genome guided assembly and variant aware mapping, and then sort of gene model work, like identifying new gene models or predicting gene models in the first place in the case of a new genome assembly. It does need downstream tools for transcript abundance estimates, and I have provided a Python script that does that in the GitHub repository that we'll come to later on. Salmon is another tool. Salmon maps or assigns reads to a transcriptome. It does this in a faster sort of quasi mapping or selective alignment way. So it's not as much of a true alignment-based mapper as HiSat. It can generate SAM or BAM output, but if the purpose of your read mapping is those BAM files because you want to use them for something like uh, gene model prediction for a new genome or a reference-based assembly, you would want to go with HiSat rather than Salmon because generation of BAM files is kind of an afterthought for Salmon. And it produces transcript and gene level quantification tables. This is really interesting because in the case where your reference genome has multiple splice isoforms annotated for some loci, like say for a given locus, there are 3 or 4 different transcripts that differ in how they splice introns in and out. Um, you can get um, you can get per transcript expression or per locus expression using Salmon. Although it does need some downstream tools to sort of put that all together for all of your samples, and that is another Python script that I've provided here. It's designed primarily for expression estimation. So if your main purpose is to quantify expression, Salmon is probably a good tool for that. And it handles multi-mapping reads, which means so let's say you have 2 isoforms of a gene that share most of their exons, but then differ in one little bit. You're going to have a whole bunch of reads that map with equal precision to both of those isoforms if they're on those earlier shared exons. Um, but what Salmon is able to do is sort of use the unique bits, the reads that do map just to one or the other unambiguously, to sort of probabilistically assign reads that do map ambiguously. So you don't get an expression summary that is sort of blended and reflects the fact that your transcripts are 90% similar at the sequence level. You actually can distinguish which are the high and low expressed isoforms. So I have slurm job scripts that implement read mapping with both of these that can be repurposed to your data that we'll come to near the end. I wanted to quickly cover these metrics for gene expression that are commonly used. TPM at the bottom is far and away the most commonly used of these now, but in older literature, you'll see RPKM and FPKM fairly frequently. RPKM stands for reads per kilobase per million mapped reads. So you basically are normalizing this against your library size in millions of reads and your transcript length in kilobases. FPKM is similar, but instead of treating reads as the unit of interest, it treats fragments, which for paired end data means if you have 2 read pairs, and one of them maps, but the other one doesn't because maybe it's in a UTR that wasn't in your reference if you mapped only on to a CDS data set. Or maybe the mate for that read pair was discarded because of quality trimming. That will count as one fragment. And then similarly, if both read pairs map, that will also count as one fragment rather than as 2 reads. Because why would you want to have pairs of reads sometimes count as one and sometimes count as 2? So this is basically a modification to RPKM that became common with the advent of paired end sequencing. TPM turns things on its head a little bit because you normalize first for the length of your genes and then for sample-wide abundance. And the really nice thing about turning the order around like this is that you get a more biologically interpretable metric because TPM literally just means transcripts per million. And so if a gene has a TPM of 10, that means that on average in the cells from which your sample came, 10 transcripts per million in each cell are from that gene. The other two, RPKM and FPKM, are not so immediately interpretable. TPM also is better for cross-sample comparison. It can get a little bit wonkier with the older 2 metrics. So in general, if you're doing something from the ground up, favor tools and methods that give you TPM over these older metrics, but you may see the older metrics in papers. And then I just wanted to quickly also touch on differential expression analysis, which is beyond the scope of what I want to talk about here. Particularly, I did try to provide some user editable scripts that could do this, but it very quickly became apparent to me that experimental designs are going to differ so much that what people actually want to compare is going to make it somewhat prohibitively difficult to have a single script that everybody could repurpose. I did want to bring three common software packages. These are all R packages to your attention as tools that could be used for this. So DEC2 is probably the most beginner friendly. And it's good for when your comparisons are fairly simple. So say you've got RNA-seq data from treated and untreated samples, and you just want to know what's the effective treatment. DE-seq is great for that. It can also do, some more complex models, like you have uh, treated and untreated, and maybe at three different time points. And you want to just see, first of all, is there an effective treatment at all? But you want to not just pool your time points. You want to do this in a time point aware manner. DEC2 can handle that as well. EdgeR and Limma get into a little bit more flexible models. So these are both a little bit more complicated, but they can implement comparisons that are a little bit more sophisticated. So I would say for simple analysis, start with DEC2. If you find yourself in a situation where DEC2 feels limiting, then start to explore EdgeR and Limma. And one really important thing I want to note about all three of these tools is that they expect a data set of counts per gene or per transcript, not something like these metrics I just went over, RPKM, FPKM, or TPM. So the tools that I'm providing you, the Python scripts I'm providing you with for this webinar, they report expression both in TPM and counts. Those count CSV files that those scripts generate would be what you want to use for differential expression analysis. How am I doing for time? I'm going to have to pick up the pace a little bit. Luckily, these next three subjects are pretty quick to go over, but actually, maybe I'll come back for questions on mapping and expression after I go over the scripts that do these things. These next 3 points, yeah, I did want to just show you also sort of what a read mapping looks like. So what I'm showing you here, this is two different views in the software suite Genius that show a mapping of RNA-seq data that you would get from loading up a BAM file across a particular gene. The green bars here are the exons, and the spaces in between are the introns. And you can see in this this sort of disjunct blue histogram at the top, this is the read depth across this entire region. And you can see the read depth drops off in the introns and then picks up immediately again. And then this just shows you where the splice junctions are, these green lines here. And if you zoom in closer, you can see the actual individual reads that are mapping on here, which is what's shown in the bottom screenshot. And just a couple of other quick uses that you might have for BAM read mapping files. You can use this for variant calling if you have population data, for example, with BCF tools. I'm not going to cover that at this webinar. I will talk a little bit about how BAM files are used in reference-based transcriptome assembly and in genome annotation in the next couple of slides. So reference-based assembly, this is when you have a reference genome, but you want to use your RNA-seq data to map onto it and generate sequences for what your transcripts are. In the event that, for example, you either don't already have gene models at all, or maybe you suspect that your gene models are weak and your samples might contain novel transcripts that aren't annotated there. So for example, let's say you downloaded a reference genome that somebody just published, but the only RNA-seq data they used to predict gene models on it was from one tissue, and your samples come from another tissue. Like say they use RNA-seq data from leaves and your data comes from roots. You might want to do a reference-based assembly just to see if there's novel transcripts or splice isoforms that were missed in their gene model prediction. So string tie is a common tool that's used for this. And I'm not going to go into too much detail. This is figure 1 from the string tie paper here that just sort of shows the general workflow. But basically, in a nutshell, this is, like I said, for reference guided transcript assembly. It takes a read mapping in the form of BAM files. So you would run HiSat2 first, builds a splice graph from exon junction evidence, finds the likely transcript paths through that, and the output is a bunch of transcript sequences in FASTA format plus abundance estimates. So this is a nice tool that can get you expression data plus unique isoforms. But if you don't need the unique isoforms, it's overkill. Um, yeah, this is just sort of the useful use cases that I mentioned already. So if you suspect there's novel splice isoforms, things like that. De novo transcriptome assembly, this is when you have no reference genome at all and you want to just assemble the RNA-seq data in and of itself. Trinity is the most common tool for doing this. And it consists of three distinct stages, which are known as inchworm, chrysalis, and butterfly. And in a nutshell, what these do is basically find paths through your reads, break those paths from the first step up into k-mers, which are just, like I said earlier, sequences of length k, where k is less than whatever your read length is. And then it builds what's called a De Bruin graph, which basically just means finding points of overlap between those k-mers and then finding paths through that graph that represent transcripts or different isoforms of a set of transcripts that come from the same putative locus. I wanted to just give a quick little shout out to UnigeneFinder, since that is software that I helped to develop in the relab. One of the problems with a Trinity-based assembly is that you get lots and lots of isoforms, which may or may not represent real splice isoforms. It's very sensitive to sequencing errors and to differences in abundance between different isoforms. UnigeneFinder basically aims to clean up that Trinity output and give you a putative set of best representative transcripts for each locus in your genome, even though you don't have a reference genome. So that's available at this GitHub link down at the bottom, and there's the paper reference there. I am going to try to go through this pretty quickly because I want to get to these scripts that you can use. And then genome annotation is another common use case for RNA-seq data. So this is figure 1 of the Breaker 3 paper, which is a really powerful software pipeline for going from an unannotated genome assembly where you just have chromosome sequences, but you don't know where the genes are on them, to providing gene model predictions. And you'll notice here, one of these main inputs that goes into this pipeline is RNA-seq BAM files, again, generated with HiSat2. So the three main types of input for this pipeline are your genome assembly, which is necessary, and then two other sort of major lines of evidence, one being a protein database, so it can look for homologous sequences, and the other being RNA-seq data, so it can look for what's actually in your RNA-seq data. You can omit this and run this pipeline just with protein evidence, but it's generally a better idea if you've got RNA-seq data to include that. So how much time do I have for going over these scripts? Not as much as I would have liked. Let's go through these quickly, though. So this is the GitHub link where you can find all of these scripts. If you Google Matt Stata GitHub, you'll find my GitHub, and then there's not too many repositories on it, and RNA-seq webinar is the one you're looking for. So what I've provided you for here is 4 slurm job scripts to carry out some of these tasks that you can modify easily for your own data, and 3 Python scripts for doing some downstream analysis. The first of these is a slurm job script that you could submit to the scheduler on a system like the MSU HPC or your own home institution's HPC. And I should also mention that most of these tools are best run on an HPC. They're very computationally intensive. And on top of that, unless you've got a Linux system or you're running, for example, the Windows subsystem for Linux, you won't be able to run them on your personal machine, most likely. So this first job script implements the FastQC and multi-QC pipeline. And shown over here on the right is just the user editable block that you might want to change here. Most important is this section down at the bottom. So you want to tell it where your FastQ files live. So in my use case here, they were all in a folder called FastQ, which was in the directory from which I'm submitting this job. You can give it a new name for where you want it to put its output. And then importantly, These are the names of the modules for the three software packages that it needs to load. These modules will work great if you're one of the MSU folks attending today. You can just use these modules as is on the MSU system. If you are repurposing this data for your own HPC, you'll have to figure out what the equivalent module names are there. And you can do that by using the command module spider and then the module name. So module spider FastQC would search for modules named FastQC. Alternatively, for folks not on the MSU system, you might just email the sysadmin and say, look, I'm looking for modules that correspond to these so that I can run a job script. And they can help you out with that. So this will run FastQC and multi-QC on all samples that it finds in this FastQ folder. Next up is Trimmomatic. So again, we have modules that need to be modified if you're not on MSU. You have to provide the directories for input and output. This is where you name the adapter file that you want to use. And so the other choices are up here in this comment. This is just sort of some runtime stuff. Then you can leave most of this alone. This trim steps you might want to modify. So these are the settings for how it actually trims from the leading and trailing edges and what the sliding window size is, and the minimum length of read to retain. This is probably an okay default to start with, but if this, for example, if this ends up throwing away like 80% of your read data, you might need to modify this. Although if this is throwing away 80% of your reads, then you may have bigger problems. This is the job script for running HiSat2. Just want to keep an eye on the time. So again, you would want to modify these inputs. So this is where your FASTQ files live. This is the name of your FASTA file that you want to use as a reference. So in my case here, I've used it on just the longest isoform per locus coding sequence only of a genome annotation for Titostromia oblongifolia. But you could modify this to whatever FASTA file you want to map your reads onto. And then just module names to modify again. Now, this no spliced alignment, if you were mapping onto something other than a CDS dataset, you would want to remove this no spliced alignment. You could just delete everything inside these double quotes for that. This is present simply because for CDS, you don't need to worry about splicing because that's already a post-splicing data set. Run Salmon, pretty similar. I don't think I need to go over too much here. Just inputs and outputs again. You can modify this fairly easily for your own data. I want to quickly go over these Python scripts to try to have time for questions at the end. And I can stay later than the hour, but I do want to end in time for people who do have to go. So the Python scripts I provided, quantify all BAM. This is for generating read expression or gene expression data from a directory of BAM files, such as you would get from the HiSat2 job script. You can call it as so, Python, the path to the script file, and then these are all the arguments. And if you call it with, instead of all of these arguments, if you call it with just -h as here, you'll get the help output that tells you how to use it. When you run it, if you include multiple threads and this progress argument, you get a little display here that tells you how it's doing as it maps the reads. And when it finishes, you get a CSV file that looks like this with all your gene IDs here and your sample IDs along here. And all of these values here, in this case, are TPM values. It also writes a file called .counts.csv where these would be all integer count values. The quantify Salmon Python script, here's how you call it. Here's the help output if you call it with just -h at the end. And what this one does that's kind of cool is it gives you both transcript level and gene level annotation. So what I've pulled out from the output CSV file here is 2 genes for which there are multiple transcripts. So this ending in 480, these are all coming from the same locus. And then this one ending in 8140 are coming from another locus. And the .1, .2, .5, and .1, .2, .5, .9, these are different transcripts representing splice isoforms. So as you can see here, it's giving you individual expression levels for each of these. The .genes.tpm.csv file is giving you the sort of pooled expression at the level of locus. So those dot numbers are removed from the ends. And then finally, there's a script for doing some quick and dirty visualization of your results. You can call it sort of two fundamentally different ways. One is just sort of with the required arguments, and it gives you a plot like this, where all of your markers are colored the same, and the name of each sample is printed on each one. So you can see, actually, I won't go over how you can see the patterns because I'll show you how patterns pop out when you include this categories argument. So in this case, I've provided a comma-separated list of category names, and what the script does is it colorizes samples based on which of these categories they belong to in the PCA. So you can see we have this kind of nice progression in PC1 from day 0 to day 1 to day 5 to day 13 and 18. So there's this real sort of effect of time, sampling time on the data. So that's the Python scripts. I haven't left a ton of time for Q&A, but I've got about 5 minutes. So I know I went a little bit quickly when I realized I was lagging there at the beginning. I imagine there's probably lots of questions, and I can stick around. And I'm also happy to take any questions that people have by email.
Awesome. Thank you so much, Matt. That's fantastic. Really, really, and for me, this is especially perfect. I just received heaps of short read data this morning, and I'm used to long read data, so this was
Awesome.
just perfectly timed.
One
There
other
are a
thing I
couple
should
of,
really quickly
oh yeah.
mention is
Hmm.
this visualize script. Um, at least on the MSU system, this script wants some Python libraries that are not in the default Python module that gets loaded. So you have to load a module called matplotlib. And if
Hmm.
anybody tries to run this and has trouble, just reach out to me by email and I can explain that to you.
Sounds great.
Yeah,
There are a few questions. So
sure.
back to the mapping tools. Luke
Yeah.
Strickland wrote that they've used Callisto for quick expression estimation.
Yeah.
I have not yet tried Salmon. So do you have experience with both, and do you prefer Salmon?
I haven't used Callisto personally, but we did. We have this Tetastromia paper out that's in current bio as of last year. That you can find if you search my name in Tetastromia. You'll find I'm not the lead on it by any means. Corinne Prado in the relab is, so I don't want to make it sound like it's my study. But I'm a co-author on it, so you can find me by name that way. When I came onto that project, the expression analysis had all been done by another member of the relab using Callisto, and we noticed that there was some sort of weird behavior where genes that we knew should be really high expression had really low expression, like C4 cycle associated genes. And I did a quantification based on HiSat and found that HiSat picked them up at really high expression. I don't know why Callisto picked them up at low expression. I'm not sure what the discrepancy was, but I have not used Callisto as a result because it gave me wonky, or it gave us, I should say, wonky data in that case. Salmon, I think, is probably the most commonly used tool these days if you want just expression-based data. Um, and the Python script that I provided is a nice way of sort of doing it across multiple samples. But yeah,
Fantastic.
I don't know a lot about Callisto. I just know it gave us some funky results, and so I've kind of eschewed it ever since. But not to say there's anything fundamentally wrong with it, but I had a bad experience.
Hmm. And Jacob Trusky asks, can any BAM file be viewed in Genius or such as accessible chromatin region mapping?
I have so I haven't tried to load a BAM file representing chromatin accessibility mapping, but it should be fine because yeah, and I don't see any reason why not. You probably don't want to try to drag the whole BAM file resulting from mapping an entire RNA-seq library into Genius. Because it's going to throw a little bit of a fit if you try to give it a 12 gigabyte BAM file representing 40,000 genes, for instance. What I like to do is use SAM tools, which is a tool that I didn't go over, but SAM tools can be used to you can basically give it a single gene or a reference of a portion of your reference genome and extract that from the BAM file. So let's say you're interested in, for chromatin accessibility, you're interested in chromosome 2 from 50 megabases to 60 megabases into the chromosome. Um, that's a pretty big chromosome, but you get my idea. Uh, you could extract that region, get a subset BAM file that's not as huge, and pull that into Genius and look at that mapping. A 1 megabase is still going to be pretty big to look at and maybe a little bit unwieldy. My general use case is mapping, gene expression data onto a genome. And then what I would do is, for example, pull out the region surrounding one gene, like I showed you in those Genius screens, to look at how the different exons are being handled.
Mhm.
But yeah, there is no reason why chromatin accessibility data couldn't be mapped because at the end of the day, all it is reads mapped to reference. It's just how you would interpret it would be different.
Mhm.
And
I
also how
see.
you would slice out your subset of interest so that you don't give Genius a heart attack.
I see. An anonymous attendee asks, are there some RNA-seq prep kits that work best? Or are there in-house kits that are typically used for certain organisms? And how do you go about troubleshooting?
There are so many different prep kits for isolating RNA that depend on your organism and your tissue and how much RNA you expect to get. Like there are micro kits for if you're extracting from, for instance, protoplasts where rather than like a bulk leaf tissue where you can get a good amount of RNA out of it. It's really going to depend on your use case. The most common quality metric for RNA before sequencing it is what's called the RIN score, which is, I believe, stands for RNA integrity number. You would get that from something like a tape station or bioanalyzer. Basically, what that relies on is the peaks in your extracted RNA size that correspond to your ribosomal RNA. If those peaks are really good and clean, and the regions around them are not a big messy smear, you'll get a good RIN score. If you've got, for example, a lot of DNA contamination, like you didn't use a DNase in your prep, you will get a lot of background and your RIN score will drop because your DNA will be sort of random fragment sizes across the whole range. So RIN scores are important. There are some use cases where RIN scores break down, though. So, for example, we do a lot of single cell RNA-seq work, which I have not even, that's a whole kettle of worms that we didn't get into in this webinar. But for single cell RNA-seq, at least in plants, we commonly use RNA from nuclei rather than from whole cells. What that means is that you don't get the ribosomal stuff. So you get, when you're isolating nuclei, you're throwing away the core information that is needed for a RIN score. So you can't get RIN scores from nuclei. So
Mhm.
yeah, the prep kit you use and how you QC it is going to depend a lot on your use case. And also, what kind of a RIN score you're willing to tolerate depends on what downstream analyses you have planned. If you need to de novo assemble a transcriptome, you might want to have a higher QC threshold than if you are just mapping reads.
Fantastic. And we're 2 minutes over the hour. Um, there are no more questions that I can see. I do
Okay,
have a quick question of my own
sure.
because you mentioned modules. When I
Yes, yes.
access a cluster, I actually have a like a conda environment that I've set up on the cluster.
Right.
So is a module kind of similar to a package that you would access on the cluster?
A
What's
module, so
Mhm.
modules are pre-installed software that's available at the admin level that they install
Okay.
for you. And essentially all that loading a module is doing is putting some additional directories in your path environmental variable, so that binary executable files in those paths can be called without having to call the whole path. Um, if you know, like if you load a module and then so for instance you load the HiSet2 module and then you run which HiSet2, it will tell you the full path to HiSet2, and you could call it with that full path without loading the module. The module is just to get it into your path variable. The reason why modules are used on an HPC system is because they frequently will have multiple versions of a given software, and they want to be able to ensure that, like, if someone's workflow depends on SAM tools from 3 updates ago, that they don't have a name clash with a newer version that's installed, so that they can load just the
Mm-hmm.
module that they need. So it's kind of like a Conda environment in that when you load it, it's sort of like activating a Conda environment in that it makes
Mm-hmm.
all of those tools available for you.
Mm-hmm.
But it's also a little simpler than a Conda environment, and it's like a top-down sort of thing that's provided for you.
Mm-hmm. I
And
see.
I should also just mention for anybody who is super, super unfamiliar with even running things on HPC, MSU's got a bunch of great sort of beginner tutorials for how to schedule jobs and how to navigate around and everything, and the module system. And I'm sure everybody else's institutions have similar. You just have to figure out where those are and how to access them.
Fantastic. And actually, one more question while I have you.
Sure.
How do tools like Trinity handle high repeat content? I study resurrection ferns, and the repeat content can be quite
Do
high.
you expect there to be high repeat content in the transcripts?
Oh, of course, we're talking about
Yeah.
RNA. Yeah, yeah.
Yeah.
Yeah. Okay, cool.
Yeah.
As we talked about the genome annotation. Yeah.
So tools like Breaker that are
Hmm.
annotating your transcripts based on a reference genome, or string type for that matter, you would probably want to use a repeat masked genome assembly
Yeah.
for these.
Hmm.
I didn't cover software tools for that, but if you do need to repeat mask a genome, a reference genome, for plants, the best tool I've found for that is called EDTA. I'm not sure what it stands for, but it implements repeat masker and repeat modeler. And I tried for a long time with a big, like a 2 gigabase plant genome assembly to get repeat masker and repeat modeler running straight up, and kept
Hmm.
getting all sorts of errors. Um,
Okay.
and EDTA managed to make it work no problem. I did have to, well, no problem is relative. I did have to tweak some of their Python code to fix it for my data set. And I did
Mm.
have to make a special Conda environment. But I can share that with you if you need to repeat mask a genome. Just send me an email.
Yeah. No, I've been working with repeat modeler and repeat masker, but I
Yeah.
I think I had heard of EDTA, but hadn't used it yet.
I
So
like
that's good.
EDTA a lot. It was for the 2 gigabase diploid genome, it was quite quick. It finished in like overnight, I think. I also had to run it on a 5 gigabase diploid genome, and it did not scale well for that. It took like 4 or 5 days, I think.
Mm-hmm.
So it was not linear how it went up from the 2 gigabase genome.
Hmm.
But under the hood, it's using repeat masker and repeat modeler,
Okay.
so it's similar.
And then
But
with the
yeah, for so
mhm,
for this purpose, yeah, you'd want to have your repeats masked out. For Trinity, it doesn't matter.
mhm. And then for the protein database, is
Yeah.
it better? I presume the database would have multiple species, or would it?
It would be more, so I think the one I'm using is either all flowering plants or maybe all
Okay.
plants. Um, I can share that with you as well if you want.
Okay.
It's
My
sort
yeah,
of
my
it's sort
question
of
was just going to be like,
domain
how
level.
broad should it be? Okay.
Yeah, no, it's you want it to be kind of pretty broad,
I see.
and pretty expansive. So I was using Maker before this, and Maker
Mhm.
would let you sort of throw in a few ad hoc
Mhm.
proteomes, and it didn't scale well to having lots of proteomes. So kind of like 3 or 4 was the max you would want to include with Maker. Breaker scales much better, and you can give it a really nice big database of of proteomes to use. Um, I should also mention that when you do this, um, it sort of adds each annotation you do to its running database. So if you're, for instance, wanting to do de novo annotations of two genomes of the same species, which I was doing, a diploid and a tetraploid accession, you would want to do the first one and then remove it because it's probably going to bias the data set too much when you go to the second
Mm-hmm.
one, having a proteome from the exact same species in it.
Mm-hmm.
So that's a yeah, something to be mindful of. But yeah, I can share anything related to this with you. I've got like I've got the fixed the scripts I had to modify on the Conda environment and everything. I think I set it up in a nice repurposable folder for the relab, so I could very easily share that with you too.
Oh, great.
Yeah.
Awesome. Well, thank you so much, Matt. And
You're welcome.
I I have this link here. I don't actually, it's my first time moderating a webinar. I don't really know where to put the link aside from the Q&A box. So I think, oh no, that goes to hosts and panelists. Where's Q&A now? The oh well, somewhere. I'm trying to put the link to the research tools and techniques webinar series for Wally. The
Okay.
next one is not yet advertised, the next seminar, um, but it will be probably very similar. Oh, thank you so much, Morgan. In the webinar chat to everyone is the link where the next talk will be advertised. We don't have that abstract at the moment, but please stay tuned. And if you're already on the email list, then you'll receive an email as well once it's announced. So
Great.
thank you so much, Matt, for
I'm
this
gonna
fantastic
quickly
webinar. Yeah.
I'm going to quickly just throw how do I change this to everyone? There we go.
Oh yeah, just
I'm
drop
going to
down.
just
Yeah,
quickly throw my email into the chat if anybody had questions or if anybody is trying to repurpose my scripts for their own use and runs into trouble, feel free to reach out.
fantastic. Thank you so much. And congrats again on
Thank you.
your new position in the fall. That's
Yeah,
fantastic.
thank you very much. I appreciate that. All right.
Awesome.
Thank you everyone for attending and thanks for moderating and fielding questions, Olivia. Been
Pleasure.
fun.
Thank
All
you,
right.
everyone.